On the demand of one of my Facebook fans, I am starting a series of lecture on the topic of anti-cancer drug. It's a vast topic, so I will not discuss each and every details related with this topic. There are numerous drugs. You don't need to memorize each and everything for USMLE and for undergrad level examination. So I will tell the basic concepts, and starting from very basic, what we will do in today's lecture is that we will understand that what is cancer and how cancer develops, and then we will understand the basic mechanism of some anti-cancer drugs, and then according to their mechanism we will classify those drugs, and throughout this process we will use some visual mnemonics so that these concepts, they get really imprinted in your mind for the long term. So I am Dr. Aizaz from MedicoVisual.com. Let's start. So what is cancer? Cancer is abnormal mass of cell whose growth is uncoordinated and uncontrolled, and it has ability to invade and infiltrate the surrounding structures as well as the distant structures, through metastasis. The term cancer literally means crab. It is named so because of its crab-like appearance. You can see that this is the cancerous tissue which has invaded and infiltrated the underlying normal tissue. It has invaded and infiltrated normal tissue, as well as it can go to distant structure. It can metastasize to distant structure. So now we will see that how normally cancer develops, the mechanism of cancer development. So there are two main types of genes that are associated, that are involved in pathogenesis of cancer. So one type of gene, they act in order to promote the cellular growth. They act as a molecular accelerator of the growth. They, in one way or another, they increase the growth of cell. The other type of gene, we call them proto-oncogenes. Then there is another type of genes. What they do? They scan the DNA, their products, they scan the DNA for any defects in the DNA, and if there is some defect in the DNA, it will act as a molecular brake. They will act as molecular brake, and they will arrest the cell cycle temporarily, so that they will allow the cell to repair its DNA. And if DNA repair does not occur, it will either cause the permanent cell cycle arrest. It will cause either permanent cell cycle arrest, or it may even induce apoptosis of the cell. So it's a protective mechanism so that these faulty mutated DNA won't be transmitted to the daughter cell, to the progeny cell. So they are, in one way or another, they are preventing development of cancer. We call them tumor suppressor genes because they are suppressing the development of tumor. They are suppressing the development of cancer. Are you getting it? So these are the genes which are involved. So what happens in cancer is that there is gain in function mutation in proto-oncogenes. I mean that the proto-oncogenes start hyperfunctioning. And when there is such mutation in proto-oncogene, we say that proto-oncogenes are converted into oncogenes. So this is the point. So when they are converted into oncogenes and they start hyperfunctioning, it means that more and more growth signals are being transmitted, and cell will undergo rapid and uncontrolled cell division, which will lead to development of such cancerous mass. And along with that, there will be loss of function mutation in tumor suppressor genes. So proto-oncogenes are hyperfunctioning and tumor suppressor genes are hypofunctioning. They are not functioning properly. There is loss of mutation in such genes. So you can understand that when there will be loss of function mutation in such genes, these genes won't be able to, their product won't be able to prevent the oncogenesis process. So as Dr. Najib says, that the cell will become like a car whose accelerator is working even when the race pedal is not being pressed and brakes have been failed, and on top of that this car cannot repair itself, and it cannot repair itself, and it has become indestructible. I mean, it cannot undergo apoptosis. So imagine how much havoc it would create. So that's how cancer develops. Now we will see that how one cell normally divides into two, because cancerous cell undergo rapid and uncontrolled division. So we will understand the basic mechanism that how cell divides, and then we will relate it with anti-cancer drugs. So let's suppose this is a cell. It is in, right now it is in non-dividing stage. It is in resting stage. We call it G0 phase or gap, G0 phase or gap 0 phase, or some people call it gap 0 phase. So under appropriate growth signal, under the presence of appropriate growth signal, this cell will plan to divide. It will plan to divide into daughter cells. So to divide into daughter cells, it has to make a copy of its DNA. It will replicate its DNA. It will make a copy of its DNA, and then one copy of DNA will remain in one cell, and other copy will remain in other cell, will go in other cell. During the process of mitosis in metaphase, they will be separated, chromatid will be separated. I won't be going into details, you know this. So first of all, the cell will go into DNA synthesis phase, and then it will go into mitosis phase or M phase, and then it will divide. But before it can synthesize the DNA, before it can copy the DNA, before it can replicate the DNA, this cell has to prepare itself for DNA replication. It has to prepare itself for DNA replication. It means that it will synthesize some proteins and some enzymes that are involved in DNA synthesis. And this phase in which cell prepare itself for DNA synthesis, this is called G1 phase or gap 1 phase. And then before cell can undergo actual mitosis, of course it will have to prepare itself for mitosis. For example, it will synthesize tubulin proteins and some other proteins that are involved in mitosis. So this is called G2 phase or gap 2 phase. So G1 phase and G2 phase. So these are the series of events through which the cell passes before it can divide into two: G1, G0, G1, DNA synthesis, or we also call it S phase. And this is also called M phase. S phase, G2 phase, M phase. And now these daughter cells are formed. Now two things can happen with these daughter cells. First of all, of course, they will differentiate. They will take some time to differentiate. Then two things can happen with them, that they may be resting, they may be in G0 phase, or again they may plan to divide further. So again they will have to, if they plan to divide again, they will have to go through all these phases of cellular division again. All these phases again they will have to go through them. So we can say that this is actually a cyclical activity. It's a cyclic activity, that this cell was sitting in G0 phase. Then if it plans to divide again, it will go into G1 phase, then S phase, and then G2 phase and M phase, and so on. So this is basically a cycle. So this cycle, it is a series of events through which the cell passes in order to undergo cell division, and this cycle is called cell cycle. So this is cell cycle. So you understood the basic concept of cell cycle. Are you getting it? Are you with me? Okay, good. It's great to know. So why I am teaching the cell cycle in this lecture of pharmacology? Do you know why I am doing so? Well, there is a reason for it. Basically, as you know that cancer cell, they undergo rapid cellular division. So most of a huge number or huge proportion of cancer cell at any time, they are in cell cycle. They are in some stage of cell cycle. They are rapidly planning to divide. So it means that we can target the cancerous cell if we target phases of cell cycle. Well, there are rather numerous anti-cancer drugs which act on different phases of cell cycle. So that's why we are understanding the cell cycle. Now, what I will do is that I will not go through each and every phase of cell cycle. Now, I will only focus on those stages of cell cycle, those phases of cell cycle on which different anti-cancer drugs act. And then we will see briefly we will see their mechanisms of action. I won't be going into details. Of course, I will be discussing major points related with them. So first of all, I will go into detail of S phase or DNA synthesis phase. In DNA synthesis phase, of course, you know that cell will synthesize DNA. Cell will synthesize raw materials of DNA as well as it will make an actual copy of DNA. So what happens here? But before discussing this, let's talk about that. You know that our DNA, it consists of a nucleotide base which could be purine nucleotide bases that are adenine, guanine, or also there are pyrimidine nucleotide bases which are cytosine and thiamine. I know you must be knowing that there is no uracil in DNA. So nucleotide base is connected with deoxyribose sugar, and then this deoxyribose sugar is connected with a phosphate group, and whole this structure is called, what this structure is called? Yes, good. It is called nucleotide. So it is deoxyribonucleotide. So it is further connected with another deoxyribonucleotide, and it is further connected with some other deoxyribonucleotides, and basically it forms a single strand of DNA. So a strand of DNA is nothing more than a long polymer of nucleotides, deoxyribonucleotides. Then there is another strand of DNA which runs anti-parallel to it, and in between them these nucleotide bases are connected with which bond? Yes, so in between them is hydrogen bond. So hydrogen bond is between them, and they are twisted around each other to form the characteristic structure of DNA which you see in the books and everywhere. I won't be discussing them in detail. But the point which I want to highlight here is that these nucleotides, and especially these nucleotide bases, this is the core component of DNA. So these are the raw materials of DNA. So cell has to synthesize these raw materials of DNA. So how these raw materials, how these nucleotides are synthesized, and how different drugs, different anti-cancer drugs act in order to arrest this process. So through a multi-step process, inosine monophosphate is formed. I won't be telling you the pathway. For that you have to study your biochemistry books. So inosine monophosphate, which is having ribose sugar, it is ribonucleotide. It can be converted into adenosine monophosphate as well as adenine monophosphate as well as guanine monophosphate. So these ribonucleotides can be formed. These purine nucleotides can be formed through this pathway. Now here is a drug which is called 6-thioinosine. It is also called 6-mercaptopurine. This 6-thioinosine or 6-mercaptopurine, it resembles inosine. So through normal metabolism in body, it is converted into inosine monophosphate. It's not inosine, 6-thioinosine monophosphate. This 6-thioinosine monophosphate, because of its structural resemblance with inosine monophosphate, it can inhibit this process. So this is one drug, one anti-cancer drug. Then we will move forward to pyrimidine synthesis. Again, through a multi-step pathway, uracil monophosphate is formed. First of all. Again a ribonucleotide. Then up till here we have synthesized these ribonucleotides. But for DNA synthesis we need what? Deoxyribonucleotides. So there is an enzyme. This enzyme will reduce this OH group, this hydroxide group. It will remove this oxygen from this hydroxide group, and this ribose sugar will be converted into deoxyribose. And deoxyribonucleotides will be formed. It will also act here to convert this guanine nucleotides into deoxyguanine nucleotides, and here also as to convert it into deoxyadenine nucleotides. Now there is a drug called hydroxyurea. So hydroxyurea, it has a hydroxy group, and this hydroxyurea, it prevents the reduction of this hydroxyl group. The hydroxyurea prevents reduction of this hydroxyl group. So actually this hydroxyurea is inhibiting ribonucleotide reductase. So if it inhibits this ribonucleotide reductase, then deoxyribonucleotides won't be properly formed in our body, and when these deoxyribonucleotides won't be formed, they won't be available for DNA synthesis. So cell cycle will arrest in S phase, synthesis phase, and this drug also causes arrest of cell cycle in S phase. So these two drugs act in S phase of cell cycle. Now from uracil monophosphate, cytosine monophosphate can be formed, as well as thiamine monophosphate can be formed. I won't be explaining this pathway, but I will be telling you about this pathway because it is clinically very relevant here. So from uracil monophosphate we can synthesize thiamine monophosphate. The only difference between this uracil nucleotide base and thiamine nucleotide base is presence of CH3, presence of methyl group. If we add this methyl group on uracil, it will become what? It will become thiamine. So it means that we can add this methyl group on uracil to convert it into thiamine. Well, there is a flying animal. This flying animal, in its mouth, it will bring this methylene group, and it will donate its methylene group as well as two of its wings. It has 4 wings, 4 hydrogen wings. So it will donate 2 of its wings, it will lose 2 of its wings as well as a methyl group in the presence of thymidylate synthase enzyme, and it will convert uracil into thiamine. It will convert uracil monophosphate into thiamine monophosphate, and of course it has now lost 2 of its wings. Now to get 2 of its wings back, it has to go to a special wingifying machinery, as I call it. It will go to a special wing attaching machinery which will attach 2 wings to this flying animal, and now this flying animal is now active with its 4 wings. It is now active with its 4 hydrogen wing. It is now active, and now this active flying animal with 4 wings, it will fly towards either serine, methionine or choline, and fetch the methylene group, and now it can again donate this methylene group and two of its wings to another uracil monophosphate molecule in order to convert it into thiamine monophosphate. So what is this flying animal? Do you know this? I know you must have heard of its name. So what is this flying animal? What? Yeah, it is. Yes, it is folic acid. Folic acid, flying animal, folic acid. So the folic acid or flying animal with two wings, it is dihydrofolate. It is dihydrofolate. It is its inactive form. To get activated, it has to be reduced. Two more hydrogen atoms has to be added to it. So this enzyme, this wing attaching machinery, is dihydrofolate reductase, which will attach two more wings to it, and now it will become tetrahydrofolate, and this tetrahydrofolate will go to either serine, methionine or choline to fetch a methylene group, and this methylene group will be attached to its nitrogen 5 and 10 at nitrogen 5 and 10 position, and now it will become N5 and 10 methylene tetrahydrofolate. It's very easy to understand, I think. So this N5 and 10 methylene tetrahydrofolate, it will again donate these methylene group to dihydrofolate reductase, and it will become it will donate its methylene group to uracil, and it will convert into thiamine, and it will lose its wings, so this cycle can continue. Now here is so you got the point. Are you really understanding it? Okay, good. So there is a T-Rex dinosaur. This T-Rex dinosaur, it resembles with this flying animal, and because of its structural resemblance, it gets stuck into this wingifying machinery of this dihydrofolate, and it will block this dihydrofolate reductase. And now this dihydrofolate, it is now crying because it cannot get the wings. It cannot get its wings, so it won't be activated, and if it won't be activated, it won't be able to fetch this methylene group from these. So now it won't be working properly. So what is this T-Rex dinosaur which is blocking dihydrofolate reductase? Do you know the name of this T-Rex dinosaur? T-Rex. Yes, it's methotrexate, methotrexate and some similar drugs, pretrexil and some other drugs which are of this category. We will discuss that later. So methotrexate is dihydrofolate reductase. It blocks this process, and sufficient thiamine won't be formed, and if sufficient thiamine won't be formed, DNA will have thiamine, DNA would be thiamine less, and DNA synthesis will be arrested. So are you really clear about this? Then this dihydrofolate this dihydrofolate, it also act here during purine synthesis. Here it fetches formyl group. I won't be going into details because it is not relevant here. Here it provides the formyl group for synthesis of inosine monophosphate. So this step will be inhibited, as well as this step will be inhibited due to this block, this methotrexate block, methotrexate mediated block. Then there is another drug, that is 5-fluorouracil. Now this 5-fluorouracil, it resembles the nucleotide base uracil. It is formed by an addition of fluorine group to uracil at carbon number 5. So 5-fluorouracil, it will be converted through normal metabolism in body into 5-fluorouracil deoxyribonucleotide. It will convert it into its deoxyribonucleotide form, and this deoxyribonucleotide form, because of its structural resemblance with uracil monophosphate, it will block this enzyme. It will get stuck in this enzyme, thymidylate synthase, and again similar to methotrexate, it will lead to deficiency of thiamine in the cell, and thiamine won't be available for DNA synthesis, and DNA synthesis will arrest in and cell cycle will arrest in S phase. So let's move forward. Now our DNA has to be replicated. Now polymerase will come, and DNA polymerase will attach here nucleotides, and it will elongate this chain. Now sometime here some beans get stuck. Now what I mean by these beans? Well there are some nucleosides that are attached, rather than these nucleotide bases, rather than being attached with ribose sugar. Normally there is ribose sugar. So rather than being attached with ribose sugar, they are attached with arabinose. They are attached with arabinose sugar. So these arabinose containing nucleotides are called, for example, cytarabine, cytidine, which is also called cytidine arabinoside. This cytidine arabinoside or cytarabine contains cytidine here, cytidine connected with arabinose, and then this will be converted into, this will further be attached with phosphate here, and this nucleotide will then this nucleotide, if it gets added here due to structural resemblance with CMP, chain elongation will be terminated, and here again the S phase, cell cycle will arrest in S phase. Then another arabinose nucleoside, that is cladribine, which resembles with adenosine. Now as the polymerase, DNA polymerase, is elongating the chain, here another enzyme, DNA helicase, it will act in order to unwind the DNA, and as it is unwinding the DNA will become supercoiled here. DNA will become supercoiled here, and now further chain elongation won't be possible until this supercoiling is corrected. Well, here topoisomerases enzymes, there are enzymes called topoisomerases which comes to our rescue. They will unwind this DNA here. There are two topoisomerases, topoisomerase type 1 and type 2. We will discuss their mechanism in detail in a separate video. So topoisomerase 1 inhibitor can be blocked, topoisomerase 1 enzyme can be blocked if we add a molecular can in its mouth. What is this molecular can? Topotecan. So topotecan is a drug which is topoisomerase 1 inhibitor. Then there is a drug which sits on the side of topoisomerase 2 and prevent it from functioning. That is topoisomerase 2 inhibitor, which is etoposide. So topotecan is topoisomerase 1 inhibitor and etoposide is topoisomerase 2 inhibitor. So again both these drugs will act in S phase and will arrest the cell cycle here in S phase. So these were the drugs that act in S phase. They prevent the synthesis of DNA and all their raw material. So these drugs are called anti-metabolites because they are involved in metabolism of, they prevent the proper metabolism of DNA and its raw materials. What are those anti-metabolites? 6-thioinosine or 6-mercaptopurine, hydroxyurea, we have already studied them, methotrexate, 5-fluorouracil, and those beans, arabinoside, cytarabine and cladribine. So these are the drugs that act on DNA synthesis S phase, and of course we also have what? Topotecan and etoposide. These are topoisomerase inhibitor. Now before the cell can move from S phase to G2 phase, and from any phase, if it has to transition from one phase to another, there are certain checkpoints. It has to pass through certain checkpoints that are mediated by what? That are mediated by tumor suppressor genes. It means that the products of tumor suppressor genes will look for any DNA damage here, and if in the event of DNA damage, the cell, they won't allow the cell to transition from one phase to another. Let's say if due to any reason, for example, if due to presence of this cytarabine, the chain elongation is terminated and there is defect in DNA synthesis here, cell won't be able to move from this S phase to G2 phase. So these checkpoints are very important in pathogenesis of cancer, because in cancer such checkpoints may be lost. There may be mutation in such genes which leads to loss of these checkpoints, and of course then cell division will go unchecked and there will be uncoordinated and uncontrolled cellular growth. Now in G2 phase, what happens? In G2 phase, the cell is being crowded with tubulin protein. So more and more tubulin proteins is being formed here, and in this crowd of tubulin protein, something very sad happens. A terrorist throws a bomb here. Oh, what's that bomb? So in G2 phase, there is a drug called bleomycin. This bleomycin acts in G2 phase. What this bleomycin does is that it generates some reactive oxygen species. It generates free radicals that damages the DNA, specifically in G2 phase. Again, we will have a detailed discussion on its mechanism in a separate lecture when we will be discussing individual drugs. Then here in M phase or mitosis phase, what happens that these microtubules are formed, and these microtubules, those microtubules which are attaching to these chromosomes, these microtubules, they will pull apart these chromosomes to each pole. They will pull apart these chromosomes to each pole, and these chromosomes will be separated into each chromatids. So one chromatids will come to here and the other chromatids will come to here. So in this way DNA will be divided into two parts. One will go to one daughter cell and the other one will go to other daughter cell. So that's what happens in mitosis. And these microtubules, how these microtubules are formed? Remember, in G2 phase we synthesize tubulin proteins. So basically these microtubules are actually the polymers of tubulin protein. So this is one tubulin protein, this is another tubulin protein. So these tubulin proteins, they are attached with each other to form a long polymer. They form a polymer. So a polymer, a long polymer of tubulin protein is actually microtubule. So there is a drug. What is this V? There is a drug which prevents this formation of polymer, which prevents the polymerization of tubulin proteins. And of course, when tubulin proteins won't be polymerized, microtubules won't be formed and mitosis won't occur. So cell cycle will be arrested here. So what is these V drugs? These are actually vinca alkaloids. For example, vincristine and vinblastine. Then sometimes we give taxes to these polymerized fibers. We give tax money to these polymer of tubulin proteins to stabilize them. We want to stabilize in we want to stabilize them in this polymerized state. We want to keep them in microtubules form. So as they say that excess of everything is bad, so excess of this polymerization of alkaloids is also bad, that if they remain stable in this polymerized phase, the cell cycle won't move further. So again, if it remains stable, cell cycle will arrest in what phase? Cell cycle will arrest in M phase or mitotic phase. So what are these taxes? These are actually taxanes. These are called taxane drugs. For example, paclitaxel and docetaxel. So these are the drugs which act in mitotic phase, vinca alkaloids and taxanes. So are you really clear about all this up till now? Okay, so all the drugs which we have studied up till now, they act on some phase of cell cycle. They act on some specific phase of cell cycle. All these drugs which act on some specific phase of cell cycle, these are called cell cycle specific drugs. But there are certain drugs which are not cell cycle specific. They can act on any phase of cell cycle. They can act on even G0 phase. They can act on even non-dividing cell. What are these drugs? Well, okay, so here is the DNA. Here is a DNA, and let's suppose here is a guanine nucleotide, and here is also guanine nucleotides. There are certain drugs called alkylating agents. These are called alkylating agents because they attach their alkyl group here with this guanine as well as with this guanine, and as they say, they cause cross-linking of DNA. They can cause interstrand or intrastrand cross-linking of DNA, and while doing so, they disrupt the normal structure of DNA. They will damage the DNA, and they can damage the DNA in any phase of cell cycle. So these are cell cycle specific drugs, and these are called alkylating agents. Examples are cyclophosphamide and ifosfamide. Then alkyl sulfonates, for example busulfan. Nitrosoureas, for example carmustine and lomustine. And some platinum compounds, for example cisplatin, carboplatin and oxaliplatin. So how can you remember the names of these alkylating agents? And how can you remember that these are cell cycle non-specific drugs? Well, it's very simple. What I think of it is that these cycles, these buses, these cars and these precious platinum compounds, these are not left at the mercy of, these are not left at the dependency of cell cycle. So these cycles, buses and cars and precious platinum compounds, these are not dependent on any phase of cell cycle to perform its function. They can act on any phase of cell cycle. So these are cell cycle non-specific drugs. And then what is this? This is rubber duck. So this rubber duck has stuck its beak here in the minor groove of DNA. So what are these? These are anti-DNA antibiotics. For example, ductinomycin, doxorubicin and daunorubicin. These anti-DNA antibiotics, they intercalates in minor groove of DNA, and again they disrupt the structure. Well, they perform certain other function as well, and ultimately what they do is all these agents, these cell cycle non-specific agents, in one way or another they damage the DNA. Some of them they also generate reactive oxygen species, and while doing so they damage the DNA. So these were the cell cycle non-specific drugs, alkylating agents and anti-DNA antibiotics. So here is another point which I want to highlight. These cell cycle specific drugs, these are specific drugs are usually given for fast growing tumors, those tumors which are fast growing and most of their cells are in cell division stage, in cell cycle stage, at some phase of cell cycle stage. While these cell cycle non-specific drugs, these are usually given for slow growing tumors because they can also target non-dividing cells. So this was the basic overview and classification of anti-cancer drugs. In upcoming lecture, what we will do is that we will study the individual class of these drugs. We will have separate lectures on individual classes of these anti-cancer drugs. But before that, in next lecture, we will discuss some basic principles related with anti-cancer drugs. So that was today's lecture. Thank you so much for watching this lecture. So much effort and work has been put into its production, so I would like you to support my work and subscribe to my channel for the latest updates and upcoming videos, and please like this video and share it with your friends, and do comments. Please share your comments that do you like this video, should I continue doing this work, and if you want if you want me to make a particular video on any topic of medical science, you can also ask in comment section. Thank you so much for watching.